Hello everyone, and a warm welcome to this webinar on the topic Statistical Aspects of Vaccine Clinical Trial Design and Analysis. This webinar is a part of series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Clive Nyaribo, Assistant Research Officer at KEMRI-Wellcome Trust Research Program, providing support to the Vaccine Workforce Development Program, together with my colleagues, Professor George Warime, Marianne Mnene, and Ravendra Navura. We welcome you to this session. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share your questions during the presentation, and we will address them during the Q&A session. We will have a few open slots for the attendees to directly ask questions to the presenter. If your internet is stable, you can raise your hand using the fireable icon. As earlier mentioned, these are series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This being run under the KEMRI-Wellcome Trust Research Program, which is a well-known health research unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbale in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision of the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. It's under this program that we have the Vaccine Workforce Development Program that aims to expand the technical know-how and capability for sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The vaccine workforce development is through 4 channels. There is 1, internships at vaccine manufacturing institutions that aims at establishing mentorship scheme that links interns with senior vaccinologists and LT/market systems and engagement with academics globally to share and strengthen experiences in vaccine R&D and manufacture and enhance career development. 2, there is a full funded master's level course in vaccinology where applicants are selected based on their direct involvement in vaccine research and their professional excellence and academic achievement. And 3, webinars like this one to area career scientists, graduate and undergraduate university students in Africa to provide information on possible career options for university students for informed career decision making. 4, we also have the Vaccinology School Engagement Program, where students from local secondary schools are invited for lab tours and career talks to spark their interest in science, especially in the vaccinology field. This program is in alignment with the African Union's platform for harmonized African health products manufacturing, ambition to increase local vaccine production to meet 60% of Africa's routine immunization needs by 2040, and strengthen the capability for vaccine research, discovery, and manufacturing in Africa. And this webinar series is part of this engagement. Today, we are glad to have Phil Bejon, who will be discussing the topic statistical aspects of vaccine clinical trial design and analysis. Professor Phil Bejon is the Director of Modernizing Medical Microbiology, co-leading with Sarah Walker. He was previously the Director of the KEMRI-Wellcome Trust Research Program, a collaborative program formed by the Kenya Medical Research Institute, KEMRI-Wellcome, and the University of Oxford. He is a clinician with specialist training in infectious diseases. Philip has an interest in the evaluation of vaccines, including malaria, yellow fever, and bipolar vaccines. He has undertaken studies of naturally acquired immunity to malaria and human challenge models, and has an interest in bone infection and osteomyelitis. Over to you, Philip.
Thank you very much, Clive, and welcome everybody. Very pleased to be talking to you all about this topic relating to the design of vaccine trials with a particular emphasis on the statistical aspects of it. I can see from the information that you filled out on joining that you're quite a diverse range of people. I mean, some of you have got quite some statistical expertise already. Some of you have had experience of clinical trials already. Others of you are more focused on the laboratory or on manufacture and don't have direct experience of clinical trials, but are interested. So that means that I am possibly going to take some knowledge for granted from time to time, and not explain a concept that needs explaining. So if that happens, if I mention something in a slide which you don't understand, or I make a quick reference to phase 2B trials or something like that, and you want to know what a 2B trial is, then please put something in the chat quickly. I'm able to see the chat. So for instance, I can see that Alenga has just introduced himself from Republic of Congo, and his interest in microscopy, the Microscopy National Network of TB. So people please put points there, and I will respond to them as we go through. If I'm feeling adventurous, I may ask for a vote at certain points, and please raise your hand to vote for a proposition. So otherwise, let's get into the talk, and like I say, let's make it interactive, and please do remind me. So I'm going to deal with two different parts. There's going to be a first part where I talk mostly about design, as informed by certain statistical considerations, and then I'm going to move on to part 2, where I deal with the analysis. And depending on how time's going, I may pause at the end of part 1 for a few questions. So the first concept I want to get across in designing a trial is that one has to take a view on at which stage of clinical development are we at. So can I just take a quick poll actually already at this point? How many people are familiar with the phase 1, 2, 3 classification? In fact, let me ask it the other way around. Can people raise their hands if they would like more of a description of what phase 1, 2, 3 means? Okay, there's enough people raising their hands already for me to get into it. So thank you very much. So look, so there's a preclinical stage, which I'm not going to talk about. So a preclinical stage involves, you know, all of the animal studies and maybe some human epidemiology and some in vitro work. Everything that is non-trial related and non-human comes in the preclinical phase. At some point, someone says, we now have a product that we want to take forward for clinical development. You know, this vaccine behaves well enough in animal models, and we have enough of a sense from the epidemiology that we've done that it could work. So you want to start clinical development, and the very first thing to do is to establish safety. And so that's phase 1. So phase 1 is typically done on a small number of volunteers who are healthy. Certainly in vaccine trials, they would be healthy. And it's done with very, very careful monitoring. And it often starts, in fact, I would say these days almost always starts with very low doses of the product, because we're nervous about it. So that's phase 1. Once one establishes safety at a phase 1, so, you know, 20, 30, 40 volunteers, and there is no safety signal, then one moves on to a phase 2, which would involve a larger number of volunteers, and at that stage is typically about immunology. So we're checking if the vaccine raises an immune response. And then following that, we move on to phase 3. And phase 3 is, as they would say, the big one. So phase 3 is a large trial. It is powered for the endpoint, which is going to lead to licensing the vaccine. So for instance, if you want to write on the vaccine label the indication, this vaccine protects you from malaria, then the phase 3 trial needs to prove that. The phase 3 trial needs to have malaria, however defined, as an endpoint, which you then show the vaccine has a certain percentage efficacy in protecting against. And so the reason for that ordering of things is that, well, phase 3 is always where one wants to get to, but it is unsafe to start with a new product vaccinating 3,000 people, say. It's, that's the power calculation we've done for severe malaria, for instance, that we need 3,000 individuals. It's unsafe to go straight ahead and do that. And so phase 1 manages the risk by looking at a smaller group of people. Phase 2 then looks at a slightly bigger group of people. So we're getting a bit more safety data. And at that stage, we can learn much more about the immunological response. And then phase 3, we get the full cohort that we need for to get a license. And it's also a stage where we'll be getting, you get safety data all the way through. And as the sample size goes up, one gets more and more safety data. So actually, I should clarify a comment earlier. There are two things that drive the sample size for a phase 3 trial. One of them is getting the indication proven that one wants to demonstrate efficacy for. And the second is getting enough safety data to say now it is appropriate to start selling this vaccine or supplying this vaccine through government mechanisms for a large group of people with some surety that it's been shown to be safe in a sufficiently large sample size. So that's why I produced this on the slides. I think you can see the balance. There's a balancing act all the way through between safety and having a powerful enough study to capture the endpoints. And you also need to think, as you go through it, about the target population. And you may not be able to immediately go to your target population. So for instance, a malaria vaccine has as its primary target population children, simply because that's where the greatest burden of disease is. However, it is considered unethical to make children the very first phase 1 trial. The reason being that children cannot give informed consent, and also that children may be less good at describing all the symptoms they experience. So it's the standard practice to start a trial in healthy adults who can give fully informed consent, who will describe any adverse event they experience in detail before then moving on to a trial in children. So I'm going to pause there to see if that nobody's giving an indication that they have not. Can people put things in the chat if they feel they haven't? All I can see in the chat now is stuff about AI assistance. And the introduction. Okay, very good. I'm going to move on. So as we talk about endpoints, we need to consider that a disease has very different endpoints associated with it. So here's an illustration of COVID, which I'm sure we all still remember, much as some of us would like to forget it. So there's a world of difference between this unfortunate soul we see on the left, who's in an intensive care bed with a presumably with a severe infection that has impacted his lungs and has meant that he needed ventilatory support, versus the person on the right who's posing to illustrate a sort of cough and or a sore throat to us. And generally speaking, for most diseases, the severe endpoints, like the one you see on the left, are the important ones that we particularly care about in public health terms. So we want our vaccine to protect people against ending up in intensive care. But on the other hand, those are the more rare endpoints. And what we see on the right was most people with COVID either had no symptoms or they had mild symptoms like the right. And yes, McConnon will share the slides afterwards, and there'll also be a recording which will include the slides on it that you can see on YouTube. So it follows that at an earlier stage of testing, one might not be, so there might be an earlier phase or a smaller trial, which one has to do for safety before going on to do the big trial. One at that stage is underpowered to detect intensive care admissions, but one could collect data about the mild endpoints. And so that may well be a very important secondary endpoint at an earlier stage of testing. It may even be put down as part of the decision making. So for instance, if a vaccine had showed absolutely no impact on a mild endpoint, it may be that at that stage, the clinical team says, well, if the efficacy is below a certain level, then we have no confidence in going on to do a larger trial. We don't think the money is warranted and the effort is warranted to do a larger trial to see if it protects against severe disease. Against that argument, and this was true for COVID vaccines, many vaccines are more protective against the severe endpoints than they are against the mild endpoints. And so taking an early decision to discontinue based on the mild endpoint might be the wrong thing to do. It might be denying the world a vaccine which is extremely useful in public health terms. So COVID is perhaps I mean, it's an example many of us remember. The phase 1, 2, 3, I mean, particularly phase 2 to 3 with COVID was very blurred because we were all in such a hurry to get on and get a vaccine into protecting people. Malaria is probably more clear cut. And so again, here we see a scene many of you will be familiar with, a child admitted to hospital with a severe malaria endpoint, possibly a form of cerebral malaria or severe malarial anemia. And so in public health terms, we really want to protect against that, but it's a rarer endpoint. I mean, the incidence, even in a high transmission site and in the youngest age, the incidence may be only a couple per thousand per year. And then on the right, we see a sort of slightly grumpy looking child amidst their friends who has, is complaining perhaps of a headache and has a bit of a fever, but nothing else to see really, and they're at school. They probably would benefit from a visit to the local dispensary and receive a course of anti-malarials, and that's a very simple thing. That can be extremely common. I mean, there are communities across the continent where there would be, where actually most children would have one or more episodes like that per transmission season. And so one can power for a trial at a very early stage against that endpoint, and that probably gives a pretty good indication. In fact, we now know that the protection one sees at that stage of testing is very predictive of the protection that one will see against severe malaria at a later stage of testing. And can I take a quick straw poll? How many people have heard? So I've explained phase 1, phase 2, phase 3. How many people have heard of things like phase 2B or even phase 2A? Yeah, so people are putting their hands up. They've heard of those, and but some people, it seems, haven't come across that terminology. So let me take a step back. In fact, let me give you a slightly more detailed breakdown of phase 1, 2, and 3. So the very first phase 1 trial done is often called the first in human trial. I.e., animals have been vaccinated and safety has been completed in animals, but the very first humans are vaccinated. Those are healthy adults. They're normally given a very low dose of the vaccine to start with because we feel that makes it safer, and that any dreadful side effect that is likely to emerge at a higher dose, we may find out a warning of it at a lower dose in the phase 1 testing. And then people often refer to phase 1b testing, and that normally means a phase 1 in a new population. So for instance, having confirmed safety in adults, one may want to then test in children. Or one might do a phase 1 test. If somebody in Oxford, for instance, is developing a malaria vaccine, the first population they undertake testing in may well be an Oxford population. Not that they're at risk of malaria, but it's just a convenient way of getting started. They may then come on to a population in an endemic country, and at that stage, that may be labelled phase 1b. Although increasingly these days we're seeing first in human studies done in endemic populations, and I think that's right. So then phase 2, there's if one can do a phase 2, so a phase 2 trial would often require several hundred volunteers to get a good spread of the immune response and to get enough safety data to go on for a phase 3. For a phase 3. There are many communities in Africa and a few actually in Asia where there's high transmission of malaria, and so one can power the trial even at that early stage for mild malaria. And so to show that there is an element of efficacy, we often describe those phase 2 trials as phase 2b. There's also a thing called phase 2a, which is an even smaller phase 2 trial, sometimes done in a non-endemic population. And at that stage, we may be doing experimental challenge. So this is where in a controlled experimental medicine environment, people are being exposed to the infectious agent. So people are injected with malaria, for instance. They're very closely monitored, and the malaria infection is treated at an early stage as soon as we detect it by PCR, and that is considered, well, not just considered, that is an element of efficacy, and that's that element of efficacy. So that distinguishes it from a sort of slightly big phase 1 or a typical phase 2, and so we might describe that as phase 2a. Okay, so I'm moving on to another description of the of endpoints, and this is about choosing the demographic group. So what you see on the left is an age graph, and it shows severe malaria, mild malaria, and asymptomatic infection. And you can see that those endpoints peak at different ages. And one has to know that epidemiology before planning a trial. So, for instance, if one starts with if one conducts a phase 2 trial in adults, there's no point in expecting to power that trial to detect severe malaria, because all of that is occurring in earlier age groups. But one could potentially do a trial based on asymptomatic infection in that older age group. And then on the right, this is a graphic showing the malaria transmission characteristics across Africa, and also showing how they change over time. And you can see that there are some areas where it wouldn't be appropriate to be doing a malaria vaccine trial, or at least it would be very difficult. One would have to do a very large vaccine trial because the endpoints are going to be so rare. So you can see that, for instance, in the modern era, particularly the areas in the Horn of Africa where malaria transmission is now quite low, and so one can't do trials there. And that takes us on to considering a power calculation. So can I take a show of hands? How many people have done a power calculation? There's quite a few, and I think I saw Benedict. Yes, Benedict, I know, does power calculations for a living, and I expect there are others. Okay, and can I now take a show of hands? So if you put your hands down, how many people have absolutely no idea how they would do a power calculation, wouldn't know where to start? Okay, so I'll give a talk from the bottom up. So the first thing to say is that there is a that a power calculation is to tell you how many individuals you need in your study to stand a good chance, and I'll qualify what I mean by good in a moment, a good chance of being able to detect a statistically significant difference comparing those who've been vaccinated with those who've not been vaccinated. And what I mean by so good chance is actually the power itself, and power is giving a percentage. So people say, I have 80% power, for instance. That means 80% of the time, based on the assumptions I put into the formula, 80% of the time I would expect to be able to detect the efficacy that I have put in. And so then in my second set of bullet points, I'm sort of starting to clarify what those assumptions will be. So first is the effect size. So what I mean by that is how good the vaccine is. So for instance, if I say the vaccine is 50% protective, that is an effect size. It means that the risk in those who've been vaccinated is 50% of the risk in those who have not been vaccinated. So that's effect size. The next is the p-value. So the p-value typically is 0.05. So can I ask, so the four of you who've still got hands up, do you mind putting them down? Because I'm going to go for another vote now. And what I want this other vote to be is how many actually, could I take a, is there anybody brave who would care to define a p-value? That'll probably get people putting their hands down very quickly. So Ndeketa, I think you just put your, Latif, I think it is, Ndeketa, I think you put a hand up. So do you mind unmuting and telling us what you think a p-value is?
Yes.
To my understanding, p-value
Yes.
lets you know that the results that you've gotten, the effect estimates that you've gotten between the vaccinated and the unvaccinated are not by chance, or the likelihood that your results are by chance. So the p-value, yes, that chance probability.
Yes, yes. So you're right, and I've got something to add. So I mean, what you have said is exactly what most people say, and it's often how most of us think about it. It is actually something slightly different in terms of the calculation. So the calculation of it actually says, if we assume that these two treatments are the same, then how often by chance will I see the sort of difference I'm looking at? And that may sound like exactly what you've just said, but I'll illustrate the difference in with a couple of scenarios. So let's say two people, we do a randomized trial of parachutes for jumping out of aeroplanes. Two people jump out of an aeroplane. One is randomized to have a parachute, the other is not. The person who wore a parachute survives. The person who didn't wear a parachute does not survive. If we calculate a p-value for that, I think it will tell us that the p-value is something like 0.6. So can I take a show of hands? How many would be comfortable with the conclusion that parachutes, that the chance that a parachute protects you there's a 60% chance that what we see is just a random result, and that parachutes are not really protective? Nobody's raising their hands. How many people would say that the explanation for that result is actually that parachutes protect you? People are now very unsure about what to think. Abonyo, thank you very much for being courageous. A few people, it's quite hard to think this one through, isn't it? And the reason it's hard to think it through is actually, I mean, it's obvious to all of us that parachutes protect you. But what is less obvious is what's going on. So the p-value is telling us, if it's just due to chance, how often, if we have a random thing, how often would we see this result? So for instance, let's take a trial of homeopathic medicine. So I'm going to, apologies if anybody here believes in homeopathic medicine. For the purposes of this scenario, I'm going to say that we all ought to take the view that homeopathic medicine doesn't work. If you had a result with two individuals being treated for illness, and what it says to us is that by chance, 60% of the time you would see this result, one person getting better and the other person not. And to extend this even further, let's say we have this homeopathic medicine which we know does not work. Let's say we do a trial of 500 people and we get a p-value of 0.01. How many people would think we've now proven that homeopathic medicine works? Does anyone want to raise a hand? So, but why not? We've got, so a couple of people who are going to say we have. I'm going to say we haven't. And the reason we haven't is that p-value is telling us that 1% of the time we will see this result by chance. And so I will say this is that 1%. So it's important you all bear in mind that the p-value is not quite telling you. I mean, for Latif's definition, for all properly, not all, for most properly powered trials, if we think of that definition, most of the time we won't go wrong. However, for those few strange circumstances, we need to remember what I've just said, that actually it's not directly calculating how likely this result is to be due to chance. And it's also important that we don't get too fixated on the threshold value. So back to the sample size calculation. So the p-value you want to collect goes into the power calculation. And there is a formula. You can do this online. So those of you who've never done one before, if you do a Google search and look up online, you will find tools that allow you to do power calculations, and you'll be able to play with it. Now, I don't mind having been delayed because I wanted to respond to questions, but I'd better pick up the speed a bit to be able to finish on time. So there's another concept here, which is about surrogate endpoints. So in a vaccine trial, the surrogate endpoint is usually the immune response. So treatment is our vaccine. we have a surrogate endpoint. This is the immune response, and we believe that the immune response then goes on to predict the final outcome. And you can see on this slide, I've also drawn an arrow between treatment and final outcome. And that is because in a phase 3 trial, we will often analyze the link between the treatment and the final outcome. And so we may power our trial based on the surrogate endpoint, we may power it based on the final outcome, but by the phase 3, it must be powered on the final outcome. And so here, I've not left myself time to talk through this, but here I've got a table showing us a collection of different diseases and the different surrogates or endpoints that one can measure at the different stages. So one can do immunity at a very early stage. I've mentioned at malaria at the very early phase 1 stage, one can well, technically it's no longer phase 1 if one is measuring efficacy against experimental challenge, but it's the same sort of sample size that one is using. And there are a few illnesses, 2 of which I've selected here, so yellow fever and meningitis C, where actually even at the phase 3 stage, one cannot get the endpoint one really wants. One is still stuck with measuring immunity because the endpoints are so rare or they're unpredictable. They tend to come in outbreaks. And so under those circumstances, one rests on the fact that the surrogate endpoint, i.e. immunity, is very closely related to the disease, and there's a lot of evidence on that. So we're comfortable licensing the vaccine on that base point. And one needs to then license it and do the post-license trials to add confidence that it's protective against the severe endpoints. Somebody's raised a thing about surrogate endpoints for DPT and hexavaccine. So with DPT, these are in the similar category to yellow fever. The illnesses are so old that there's a lot of literature on the immune responses that are required for protection. And so people use, if you're doing a new trial for a new dosing regimen, for instance, of either of the DPT vaccines, one does it based on the immune response. And then we've got enough confidence in relating the immune response to outcome to be able to license new vaccines based on that. What one would do then, though, is a non-inferiority trial, and I'm going to come to that. In fact, I'll skip over that slide. I'm going to come to non-inferiority as a point in a moment. This is a particular issue for vaccine trials. So I'm sure you've all heard of blinding, and that the proper way of doing a randomized controlled trial is to blind, so that people have so that neither the investigator nor the patient nor anyone else connected with the conduct of the trial knows which treatment people have been allocated to. And that's incredibly important because we know that there are lots of subtle psychological effects for patients or volunteers reporting or not reporting effects based on a placebo effect. And we know that nurses, doctors and others involved in looking at endpoints are can be very susceptible to recording data in a way that they've been suggested to, because they believe the vaccine has worked. So this probably isn't really malaria, for instance, as somebody might be induced to think. However, with vaccines, we often struggle to fully blind it. So blinding often comes from the small molecule world, where people are giving tablets. And what they do is the tablets come out of the factory, and the tablets have a capsule around them, and it's the same capsule for the placebo and for the same tablet for the placebo and the same capsule for the placebo and same capsule for the active drug. And then it's chipped off, and it's only unblinded at the end when the person who kept the code attaches it to the patient records. We often can't do this with vaccines because the vaccines are in different volumes. We're often more restricted with the manufacture process so that our vaccine comes bottled in a certain way. And so we can't do double blinding in the vaccine world. So what we often do is a thing called masking. So masking means that there is a so the study team who are involved in most of the work don't know about the allocation. The volunteers don't know about the allocation, but there is a vaccinating team which do know. So when somebody is enrolled, the vaccinating team are then told what they should do, you know, whether it's the active vaccine or the control vaccine. They then draw up the vaccine into a syringe. They will often then put masking tape around the syringe so that the volume of liquid and the color of the liquid is not clear. And then that is passed through a hatch then to another member of the study team who is in contact with the volunteer and gives the vaccine. So we can't really call that double blind. It's a form of masking, but it protects the conduct of the trial much better than if the study team knew about it. Now, I can see somebody's asked a question about multiple endpoints without flating to one error. So I'll deal with that, but this takes us back to the p-value question. And actually a thing I ought to have said. So we generally fix a p-value threshold as 0.05. I'm sure people are familiar with that. And there's a story behind the fixing of that p-value. I think it's a story, but it's a useful one. And that is, so it was Fisher. Fisher was the statistician who decided that 0.05 was the right threshold. And a lot of Fisher's work was agricultural science. And it takes you roughly a year to do an agricultural scientific experiment because that's the length of a growing season. And he thought in those days that he could do 20 experiments during the course of his lifetime, during the course of his career. And he reckoned that if he made one mistake in his career, that was okay. So a p-value of 0.05, so 0.05 times 20 is 1. So that means he has an odds-on chance of making one mistake during his career. So that's how it's fixed. And the reason that story is useful is that it shows us just how arbitrary fixing the p-value is. So today, when many of us can expect to be involved in work generating, you know, many more than 20 studies during our careers, actually it's not appropriate really to stick with the 0.05 threshold. I think we ought to be more confident, particularly if there's a vaccine that's going to go, you know, a single trial with a p-value of 0.04, and now we want to vaccinate millions of children. I don't think that's appropriate. We need more confidence these days. And this also takes us on to the question about multiple endpoints. It follows, if I include 20 endpoints in my trial, and I fix a threshold of 0.05 to them all, it follows that just by chance, one of those is going to be an apparently statistically significant result. And this then links back to the importance of being very precise about the definition of p-values like we were earlier. So to protect us against that, the way of handling things is that when one writes an analysis plan for an efficacy study, one picks the primary endpoint. The primary endpoint is where we are staking all of our chips, to use a gambling metaphor. And for instance, in a malaria study, we may say, well, we want to protect against clinical malaria in all children. That's going to be our primary endpoint, and that's where I attach my 0.05 threshold, although bearing in mind that it probably needs to be substantially below 0.05 to give confidence. All the secondary analyses are contributory to our thinking, perhaps, but should not contribute to the major decision taken with that trial. If we want to use them, or we want to undertake multiple endpoints and say that actually all of these are important, then the only way of handling that is to divide the 0.05 threshold between them. So for instance, say I had a vaccine and I said, I think this vaccine is going to be protective against clinical malaria, and for some reason I think it might also have an impact against hepatitis B. And I'm going to measure hepatitis B acquisition in this trial. I should really, if I want to make those endpoints co-primary, then I should assign a threshold of 0.025 to the malaria one and 0.025 to the hepatitis one. And do you see that's still protecting us? So because they're together, they add up to 0.05. That's bringing us back to the position where we think we've got a 1 in 20 chance of making a mistake. Because we've got a 1 in 40 chance of making a mistake for each endpoint. So together, that gives us 1 in 20. So I hope that makes sense. That was Mwangangi who asked that question. I hope that makes sense. So if I summarize this section of the talk, the important thing as we design our trial, the important thing to frame the question is, what is the endpoint that I really need to eventually, you know, the last phase 3 trial, what endpoint do I need to get to? And so to get there, which participants do I need in terms of the demography? How many of them do I need? And what surveillance am I going to need in order to get all of that? And then, having thought that through, I need to ask myself, is this trial going to be practical? You know, can I get the sample size that I want to get? And how can I devise a blinding strategy that makes it practical, which might be masking? And then, having done that in very rough outline for the phase 3 trial that I want to get to, then I ask myself the question, is it safe to do the trial I want to do? And the answer will always be no. I mean, it is never safe to go ahead and do a phase 3 trial as the first in human study. And so then I walk backwards from that. Well, what trial do I need to do before the phase 3 trial? And then what trial do I need to do before that? And so then I walk backwards to my first phase 1 trial. And I will say there is absolutely no point in doing a phase 1 trial if one has no idea where one's going in terms of a phase 3. If there's no phase 3 and there's no licensure strategy that applies, then it's better not to waste everybody's time. But probably more likely, thinking through the phase 3 trial helps one design the right phase 2 and the right phase 1 trials. Which population do I need to establish safety in first? Which immunological responses do I need to demonstrate are up to the surrogate markers that I have, and so forth? And I can see I've only got Clive, am I right that I've only got 10 minutes left?
No, we have around 40 minutes.
4 minutes left.
Yes. Yeah.
Okay, so
40,
look,
yeah.
oh, 40. Oh, okay,
Yes,
in which
yes,
case I
yeah.
can go on to part 2. Okay,
Mm.
very good. So this is moving on then to the analysis of the study. Actually, before I rush, now that I calm myself down, realizing I've got a whole 40 minutes left for this bit, does anybody want to ask questions about things that I've said? Yeah, McKenna? McKenna, you seem to be on mute.
Yes, thank you. Thank you so much.
Yes.
I wanted to ask, do power calculations vary according to, let's say, the disease of interest? For example, if you have like a rare disease,
Yes.
that, you know, how do you calculate power as opposed to, let's say, a very common disease, for example, like influenza, or even the severity?
Yeah.
Maybe some diseases are very severe and others are not as severe.
Yes.
Do power calculations vary?
They absolutely do. So the best power is when you have close to 50%. We often say that the best power is, you know, that the more common a disease is, the more power we have. But actually, if nearly 100% of people are going to get the disease, then it can be quite tricky. You know, you can end up, in a mathematical sense, you can end up with low power at that stage. But let's assume we're not there. So the closer to 50%, the more power you have. The closer to 0%, the less power you have. So for the rare disease, you need a bigger and a bigger trial. There's another complication which I perhaps should mention to you all, and this is often overlooked. If your disease is one that can easily be misdiagnosed, then that also reduces your power. So for instance, let's say we're doing a COVID study and there's lots of coughs and colds in the community, and we can easily mistake a cough or a cold for COVID. When we do our study, we will mistakenly include lots of things that aren't COVID as part of the endpoint. The vaccine cannot protect against those. So a COVID vaccine, for instance, can't protect you against flu. And so it follows that because we're adding endpoints to both the control group and the active group, we're losing power. And even worse than that, we're biasing our estimate of efficacy. So for instance, let's say that a COVID vaccine is 100% protective. So if we're only looking at COVID endpoints, we're going to see 100 COVID endpoints in the control group. We're going to see 0 COVID endpoints in our vaccine group, and we'll correctly conclude 100% efficacy. However, let's say that what we call 100 COVID endpoints are in fact 50 COVIDs and 50 flu. So with our 100% effective vaccine, actually, sorry, 100% effective is a bad one to choose. Let me choose 50% effective vaccine. So with our 50% effective vaccine, we're going to prevent 25 of the COVID episodes. We're going to prevent none of the flu episodes. So in our vaccinated group, we're going to see 75 endpoints. In our unvaccinated group, we're going to see 100 endpoints. So we're going to say that our vaccine is 25% effective. It wasn't, it was 50% effective. But we've diluted so the surveillance and the disease and the definition of the disease as well as the disease itself are absolutely critical. And McKenna, to enlarge on your point, it follows that if the disease is very rare and the things that we may confuse with the disease are very common, then we're in terrible trouble and we actually almost can't do the trial. Somebody,
okay,
yeah, thanks for getting. So somebody has also raised proof of concept trials. Sometimes the sample sizes are very small. How do we get good statistics? You can't, I suppose, is the brutal answer. You can't get them for the endpoint you really want. So if you've got 20 adults with malaria, you cannot get good statistics on malaria episodes. You simply can't. But when you call it a proof of concept, it often means that you've got some other surrogates that you're introducing. So for the case of malaria, that may well be the challenge study. So I can't identify febrile malaria in the real world with these 20 adults. What I can do for my proof of concept is I can inject them all with experimental malaria, and I can measure the growth rate of that experimental malaria by follow-up blood tests, and I might be able to demonstrate that my vaccine reduces the growth rate. So I would say then that I have proof of concept. I have proof of concept that this vaccine does something useful in a human being in vivo, and that might give me confidence to then go on to the phase 3 trial, despite my small size, because I've done something, you know, that allows me to get lots of endpoints in my smaller trial. And then Chinaza has a question that we shouldn't necessarily stick with 0.05 p-value and should aim for a lower. Do I have general recommendations? Good point. I think it is case by case. I think that power calculations for efficacy at early stages of testing will often still be done with a 0.05 threshold. And I think that's appropriate because often at that stage, one is not saying, I'm going to go on and give this vaccine to millions of children. What one is saying is, I'm going to go on to the next stage of testing. So let's stick with the proof of concept example that I gave Robert a moment ago. So that proof of concept, I might well power that at the 0.05. And let's say I get a 0.04 or 0.05 p-value. I might well say, okay, I can't have confidence that I can give this vaccine to the world, but I do have confidence this is an interesting thing to take forward to another stage of testing. And in practice, by the time I get to a phase 3 trial, because of the size that I need to do a trial at in malaria to get safety data, I will often have an extremely low p-value for my efficacy against the very common endpoints. And by the way, I would say even if my proof of concept trial, say I get a p-value of 0.08, so it's above 0.05, I still might well feel that this is an interesting, say I've protected, you know, my vaccine has protected a reasonable number of volunteers in my challenge arm, and by bad luck, in my control arm, I ended up not infecting a few volunteers. You know, the challenge didn't work very well. So overall, my p-value is 0.07 or 0.08 or something. I still may well say I have enough confidence in this product to go on to the next stage of testing. So I do agree. I think it is more case by case to answer Chinaza's question. A lot of that case by case will be done post hoc. The setting up and the formalities of it will often still stick with a 0.05 p-value. And Prosty is asking about estimands. I am going to, let me get through a few slides and I'll come on to estimands. And I'd better get moving through these additional slides now. So let's get so this is a consort diagram. So just about all clinical trials should have a consort diagram as their figure 1. And this is a typical one. I mean, this is a trial we did in Kenya, where we randomized volunteers to the RTSS vaccine versus rabies vaccine as controlled. We chose rabies vaccine as controlled because we felt it was a way of giving some benefit to the children who were not randomized to get the malaria vaccine. And what you see underneath then is a timeline for how many got the different doses, how many entered extended follow-up, how many completed the study, and how many dropped out with the reasons for why they drop out. And this is fundamental to one's understanding of the trial results, because if you lose lots of follow-up, it raises the spectre of bias, because you're at the stage that you randomize, so right up the top here where we randomize our 447, we have absolutely beautiful even balancing by our randomization between the two groups. Once people have had the vaccine, they are changed. And if they drop out, especially if they drop out for vaccine-related issues, then that's trouble. So for instance, say we see here, after the first dose of vaccine, 50 or 60 children are withdrawn from the study. That would be a real worry, because those children may have dropped out because of a vaccination adverse event, side effects that they didn't like. And so firstly, that now means our statistics on the safety are biased because we're missing important events. And secondly, those children may well be different in ways that we don't yet understand from the children who did not have side effects. And we are estimating our endpoints now only in those children who didn't have the side effects. And that may bias our endpoints. And I'm going to come back. This concept of estimand is a very interesting one, and I'm going to come back to that concept of bias when I deal with it. So there's the other thing that everybody should have is a baseline characteristics that shows you, the mean age and sort of sex and so forth. And that gives us a sense of whether we were unlucky. And despite the fact that I say our randomization, guarantees balance, I mean, of course, it can't completely guarantee balance. We can't guarantee balance. Sorry, and we may be unlucky. We may end up in a situation where just, by bad luck, we've got more. We've got older children, for instance, or children who live further away. And this gives us an indication that that hasn't happened. I've said no p-values. And the reason I've said no p-values is that if you think back to our earlier discussion about what our p-values are telling us, the probability that any imbalance is due to chance. Well, we've randomized these people. So the only explanation in the universe for any imbalance is chance. So it makes no sense to do a calculation to decide whether the imbalance is due to chance. We know that whatever we see is by definition due to chance. So p-values shouldn't be included here. Okay, so here we come on to our estimands. So firstly, I'm going to cover according to protocol versus intention to treat. And sometimes according to protocol is called per protocol. Now, can I take a show of hands? How many people understand the definitions of those terms, per protocol and intention to treat? A few people do. How many? Only a few people, actually. So how many people would want an explanation? If you put your hands down, those who know about it, put your hands up if you want an explanation. Okay, there are some new hands going up. Very good. Okay, I shall I'll give you an explanation then as to what the what it means. So intention to treat means that we intended them to be in the trial. They may have dropped out. We may not have got all of their data. Anything could have happened, but nevertheless, at the start, we intended to treat. And so that is basically the point where we enroll someone into the study. And well-designed trials will try and put randomization at the point of intention to treat. So often what we do with our trials is that we tell people, okay, you meet the eligibility criteria. come on, this is your date for vaccination, and if you come on that date, we'll randomize. And so that avoids people sort of dropping out after being randomized, which is very messy. And that brings intention to treat right at the point of randomization. And it means that we have confidence in equipoise. Equipoise was the wrong word. We have confidence in balance of the two randomized groups at that stage. Sometimes people talk about that as being the real world. And I don't think that is such a good argument, actually. I think the better way of thinking about it is that it's close to, because the fact is that any clinical trial is not really the real world. I mean, you can design pragmatic trials deliberately to be more real world like, but most of the time it's going to be phase 4, particularly in vaccinology, which is real world. So according to protocol or per protocol means that somebody has met certain standards set out in the protocol. So often in vaccinology, we would say somebody needs to have all of their doses. That would be per protocol. And the reasoning in saying that's per protocol is that we say, well, look, I know that my vaccine, if you only have one dose, I know you don't get a good immune response. And so what's the point in including those people in my trial? Or people, we might say people need to complete at least one follow-up visit to meet the according to protocol, and various other things. Now, the big problem with according to protocol is that these are post-randomization events that determine according to protocol. Whether someone comes back for dose 2 or for dose 3 is post-randomization. And at that point, there is a risk of bias, and we can no longer be completely confident that our two groups are equal. We can show our consort diagrams and say, look, there are only 5 children who dropped out after dose 1 or whatever it is. And that's very helpful, but it doesn't completely deal with it, because what if all 5 of those children were going to go on and get an endpoint? And so for that reason, per protocol and according to protocol analyses have become less and less favored by statisticians and people who look at clinical trials, with one exception, and that is this concept called estimands. So an estimand literally is a quality. So let's talk about vaccines here. It's a quality of the vaccine in terms of having a particular effect in a particular group, and it's defined in those terms. So we might say our estimand is in the children who have 3 doses, and we may then specify some other thing about their follow-up that we will see we estimate protection in that group. In order to try and counter the bias, what statisticians then set out in advance of the unblinding, often in advance of the trial starting, is how they will impute the missing children. So we'll say, okay, there are 5 children who went off and we missed them. What other data about them that we have will we use to impute what happened in those children? And so one tries then to get an estimand constructed from the data that we have. So it follows that this process is really quite involved and requires someone who's done it before and has a very clear plan for doing it before the trial ends. And it means still intention to treat is going to be prioritized and is going to be the safest way of doing it. There's one exception. The one exception is a non-inferiority analysis, and I'm going to show you because I think somebody else asked about non-inferiority. So before I finish, I'll talk through non-inferiority and what it means for DTP and why ITT wouldn't be the right thing to do for it. Zanetta, I can see you've got a hand up. Do you want to ask a question? Zanetta, you're muted if you're asking.
It was a previous hand up, sorry.
Oh, okay, no problem at all. And somebody's asked, what's the best way to define enrollment after randomizing? No, so the way I would do, the way I would always try and design trials is that enrollment, randomization, and getting the vaccine is all, or the first dose of the vaccine is all at exactly the same moment. Because it makes your life so much easier with all of these complexities. And I would really try and avoid, if you enroll and randomize somebody at a sort of post-screening visit and tell them to come back next week for their vaccine, many won't come back. And then you're in a real mess in terms of describing your consort diagram and so forth. How do we account for participants randomized but not IP administered? Can they be included? Yes, they must be in the ITT. If you have randomized somebody, that is an intention to treat. It might follow that one needs a definition of randomization, by the way. So the fact that a statistician assigns the electronic equivalent of an envelope that you're going to open doesn't mean that they've been randomized. Okay, it's the date. So they turn up to the clinic, and at that point you open the envelope. That's when they've been randomized. And it's important. And the point is, that's the stage. At that point, you've opened the envelope and it says rabies, not malaria. That's the stage where somebody has been randomized. And so, I mean, it's very unusual, but if somebody was at that point to say, well, actually, I've changed my mind, while you're drawing the vaccine up, then you have to still include them in the intention to treat group. Okay, so we've discussed primary and secondary analysis a little bit already. And really only the primary analysis and primary endpoint is what matters. The secondary analysis can be useful. It could guide the vaccine developers in how they think the vaccine may or may not be working. It can guide them in terms of what you might need to do next, but it won't help you in terms of licensing. The regulators won't consider any of those as being of any value. And I've dealt with these points about what a p-value really is and multiple comparisons. I've dealt with them earlier. Somebody's got, can it make sense to define additional populations for immunogenicity analysis, e.g. in vaccine trials with 2 primary and a 3rd late dose, since the 3rd dose wouldn't be needed to assess immunogenicity after 2 doses? So the way I would do that is not by defining different populations. What I would do is I would take post primary immunization bloods. So what would often happen is people would have dose 1, blood test, dose 2, blood test, dose 3, blood test. And so you would assess in that same group what the impact is of additional doses. If you want to look at the duration rather than just the peak response, then you can't do that. And especially if you want to look at efficacy, then you can't do that. And in such a case, what I would do is then formally randomize people to different arms. So I would have one population where you're going to give 3 doses, another, one randomization arm where you're going to give 3 doses, another randomization where you're going to give 2 doses, and another randomization where you're going to give 0 doses. And then you would compare the 3 with each other. I've actually already covered that a bit. So interim analyses. So I guess people are probably familiar with this going on, and occasionally you hear. So, for instance, a little while ago, there was an E. coli vaccine trial in the UK, which was stopped based on an interim analysis. And there are lots of reasons one might stop a vaccine at an interim analysis. One might stop it because the vaccine works. You know, you've already seen enough endpoints, and it's time to report that you've seen these endpoints, and people can get on with the next stage of testing. The other reason for stopping is that one might have decided it's not safe. And then there's a third reason for stopping, and that may be that for what's called futility, i.e. it's pointless to carry on. So what they mean at that stage is that that we're measuring essentially sort of 0% efficacy or very low efficacy. We know that even if we carry on the trial for another year or two and collect more endpoints, it's very unlikely that that's going to turn 0% into, you know, 70, 80% or useful efficacy. Now, if you want to stop for efficacy reasons, for demonstrating that you've already shown efficacy, you need to be aware that by doing an interim analysis, you're basically doing an interim analysis. Sorry, by doing your interim analysis, you're basically doing multiple analyses. Because, for instance, what happens if we do 20 interim analyses? One of those is going to be significant just by chance. So it means we can't apply the 0.05 threshold. And there's this thing called the Habital-Peto rule, which is a very useful posh name for simply applying a low p-value threshold, so 0.001. That is not the rule that would be applied for safety. So let's say that we're using a treatment. Let's stick with the malaria vaccine example. So we may or may not have seen any indication of efficacy yet, but we are seeing severe muscle reaction. It's an intramuscular injection, and we're seeing severe intramuscular reactions compared with the other vaccine. That safety signal will be a difficult one to interpret, but it would not be appropriate to say, well, look, we absolutely need to prove beyond all reasonable doubt below the Habital-Peto 0.001 threshold that this vaccine has a serious safety concern. It would probably be appropriate to pause vaccination and consider very carefully whether to stop the trial at a much higher p-value. And then futility analyses, I'm not going to go into those, but futility analyses again would have a lower threshold for concluding that that continuing might be futile. So now I'm going to get into a slightly more technical aspect of the choice of analysis. And I'm aware from the answers I've got so far and responses, I can see that there's a fair number of people who wouldn't particularly benefit from getting into the details of this. So the message I want to give to you is that the way endpoints occur influences the choice of statistical model. So the simplest way that an endpoint occurs is it's a yes or no answer. You know, the study participant either dies or doesn't die, for instance. And in that case, there's a simple model that will allow us to compare proportions. And a logistic regression analysis is one which is suited to an outcome which is either yes or no. And that will allow you to adjust for various other factors. Now, there are some other events. We talked earlier about the sort of event where we might expect 100% of the volunteers to get this event. So there are some malaria transmission situations where one might expect 100% of the volunteers to get malaria. Under such circumstances, the time taken to get that episode is an indication of how much risk someone was under. So for instance, somebody might get receive the vaccination and next week they have an episode of malaria. That person is probably at higher risk than somebody who nearly makes it to the end of a year's surveillance and then has an episode of malaria. And so the time to event is the sort of information that can power a survival analysis. Also, in such a situation, the number of events might be useful. So somebody might have got malaria, but during the year, have they had 5 episodes of malaria, or if they only had 1? And there's an analysis called a Poisson regression model and other related models, which allows one to analyze that. So the choice of model has to fit the distribution of the endpoints. And here's an illustration of a Kaplan-Meier plot, which is illustrating time to event. And you can see that the red line, which is the line of children who received the rabies vaccine, is going up much more quickly, showing that the time to event was faster in that group. And a survival model will allow you to calculate a percentage efficacy for the risk demonstrated by that line. I'm going to skip over some of these because I'm running out of time. But what I do want to cover is this. So this came up, I think somebody asked about DTP vaccines. So all of the trials I've been talking about so far have been things called superiority trials, i.e. we have a control vaccine which doesn't work, and we have a new product, and of course a new product has to work better than control. I mean, otherwise we're saying it essentially doesn't work. But there will be some situations like the DTP situation where there already is a vaccine. We have quite a lot of confidence that this vaccine works, but we might want to do a new trial because we have a new product which either is cheaper, has less side effects, or perhaps just is a is a more assured manufacture process, and we need more vaccine in the world. So under those circumstances, we're doing what's called a non-inferiority trial. In other words, we're saying this new vaccine is valuable because of its side effects or some other property. And of course, if it's inferior to the existing vaccines in terms of its protection or its immune response, then I don't want it. But if it is not inferior, then it's good for me and I'll use it. So hence non-inferiority. And so what I'm showing in this top plot where superiority is, this is what we're trying to show. We're trying to show that there's an estimate, which is a positive number, 30% or something, and where we have confidence intervals that exclude 0%. Therefore, I'm going to say, I do not believe this happened just by chance. I believe that my vaccine is superior to control or to no vaccine. With a non-inferiority trial, what I'm trying to show is something like this, i.e. that is the point estimate is 0% or something thereabouts, and the lower bound excludes some margin that I've set. And you have to set a margin, because you can never prove that something is 0%. You can only prove it within some certain bound of uncertainty. And so what one has to do is say, well, look, if it was, I have to define what I mean by non-inferiority. So if this vaccine was 10% worse, I'm going to call that a clinically important worse value, and I'm going to say I don't want it. Or one might say 5% or 20%. And it's helpful to have some basis for deciding that, some calculation that, so for instance, with DTP, let's look at diphtheria. With diphtheria, one might say that herd immunity will be at risk if the vaccine is 10% worse than the existing one. Therefore, my non-inferiority margin is 10%. And so that's how I'm going to power my trial. So that's non-inferiority, to bring a new vaccine into an existing vaccine market. So intention to treat came up also as an issue, and definitions of it and so on. And we said that for superiority trials, intention to treat is generally going to be the right way of looking at it. There's a problem with intention to treat in non-inferiority trials, and it relates to non-specific endpoints. So let's say I do my non-inferiority trial with my new diphtheria vaccine, and I want to measure an immune response. And it turns out that, let's say, half of the children in the study, unbeknown to me, had already had a diphtheria vaccine. So they've already got a diphtheria response. I might say, well, that's not according to protocol, because I wrote my protocol about only recruiting people who didn't have a diphtheria vaccine. And these people have now had a diphtheria. So they're out of my according to protocol analysis, but they are in my intention to treat analysis. So if I think that intention to treat is always the right way to analyze, I have a problem, because let's say my vaccine is substantially worse. Say my vaccine, in fact, let's say my vaccine doesn't work at all. If I include these 50% of children who've already had their diphtheria vaccine, I'm falsely led to think my vaccine worked. So, okay, it's only going to be 50% compared with, let's say, 90% in my in the old diphtheria vaccine that I was confident of. So I'm still going to come to the right conclusion that the vaccine has a problem. But do you see how what would have been a 100% negative value has now come up to a minus 40% or 50% negative value? And so with less extreme examples, I might turn something that would have been minus 20% into minus 10%. So I might falsely conclude that the vaccine is non-inferior, or conclude that I've shown non-inferiority when in fact I haven't. So it means that one does need to do per protocol analyses for non-inferiority trials. I can see, so somebody's asked a question about immediate and delayed vaccination groups. Yes, so I guess what you're driving at is that the delayed vaccine could result in it gives you a different timeline, so that event monitoring starts at a different point, and then you have all sorts of problems. I mean, somebody might drop out before they get their delayed vaccination. And so the way I would do a delayed vaccination group, if it was sort of, one of these day 0, day 30, day 60 for one arm, and the other arm has day 0, day 30, day 180 or something like that, I would start the delayed group much earlier, so that the last vaccination comes at the same time for both groups. And that way you have comparability in terms of the post-vaccination surveillance. Because you have a real problem if in one group you're recording endpoints before they've completed their intervention. It makes it very hard to analyze whether there's any benefit. And I think this is going to be the last thing I tell you about surrogate endpoints. So there's, I mentioned this right at near the start, that one wants to, in a model, one wants to analyze treatment versus final outcome, and also treatment versus the surrogate endpoint. And often one wants that surrogate endpoint validated so that future vaccine development can be confidently based around that surrogate endpoint rather than the final outcome. By the way, let me just briefly mention, there's a big confusion in the literature as to what this is called. Some of the literature calls it a surrogate, some calls it a correlate, and people use those words with very specific definitions, sometimes in the opposite ways round. So I think in vaccinology, one should always define what one means by a surrogate or a correlate. So this is what I mean at this point. I'm saying it's something that predicts the final outcome. And there's a thing called the Prentice criteria, which says that if you want to validate a correlate or a surrogate for future use, all of these 4 things need to be true. So I suppose obviously your vaccine must be associated with protection, or the whole thing's a non-starter. The surrogate or the correlate must be associated with protection. And obviously the correlate must be associated with vaccination. It must be a consequence of vaccination. And then this thing, which I'm going to illustrate to you in a moment, it says that in a multivariable model, the correlate is an independent predictor. The vaccination stops being a predictor once a correlate is included. So before I show you the example of that, let me take you back to this diagram. So what that is saying is that for a surrogate that we measure in the laboratory, so all of these lines must show a statistically significant relationship. And that's what I've said in words with my 3 bullet points on the slide that followed. But then there's also one other thing that must be true. So this is my illustration of it. So the vaccine is roughly, so that's a hazard ratio of 0.54. So that shows you that it's nearly 50% protective. So that's criteria 1. If I add the cellular response, so the cellular response is associated with protection. Fine, that's criteria 2. I haven't shown you that the cells are associated with vaccine, but take that for granted. That's criteria 3. But the vaccine, in a multivariable model that includes both cells and vaccine, the vaccine is still doing something. Do you see? We've still got a bit of a p-value there, and we've still got a bit of a hazard ratio there. So that does not meet this 4th criteria. What does meet the 4th criteria is this one, where we introduce antibodies. So antibody is associated with protection. And you can see it's more associated with the protection than cells were. But once we have antibody in the model, vaccines are no longer associated. This is a non-significant result, and the point estimate is quite weak. It's only barely protective. So what that is telling you is that once you have a model with everything in, this to that, i.e. treatment to final outcome, is no longer important. So it means that everything that treatment does goes through this surrogate endpoint. So that is telling you that this surrogate endpoint is gives you confidence in the vaccine working. That if you measure that surrogate endpoint, you can then predict whatever protective efficacy I might have expected from the vaccine. If it gives me the same surrogate endpoint as other protective vaccines, I'm going to bet on it having the same impact on the final outcome. So this is quite subtle. And I think for those of you who are not statisticians, you will always have a statistician working with you on this. But I think it's important that you appreciate this, that there are circumstances where a vaccine may be associated with an immune response. that immune response may also be associated with protection, but the statistician, after doing this analysis, is going to say to you, I do not think this is a surrogate marker, or what some vaccinologists would call a correlate of protection. And that's a bit of a tough message to receive, but it's important you understand that there is a theoretical and a practical basis for that. So there we are. That's my summary. When you write up stuff, always have a consult diagram in a table 1. Understand the difference between per protocol and intention to treat, and how you should use them, and how you should generally prioritize intention to treat apart from those few other circumstances that I've described. Bank on the primary analysis rather than the secondary analysis. Select an analytical model that suits your endpoints, and beware in the complexities of the analysis of correlates of protection. And that's it. So thank you very much, everybody who's stayed the course. I think I've run a bit longer than you might have expected. We've taken questions and things all the way through, but I'm happy to stay on and answer any additional questions that people have that haven't been dealt with so far.
Thank you, Phil, for such an interesting presentation. Might take just one question if there is any. Then we close, but the chat will be open for any questions. You can type your questions even after the webinar has ended. If there is anyone with one question. I believe
no, I think people probably had a chance as we went through. Yeah.
Sure. Maybe you could make your final remarks, then we close.
Oh, no, there are a few coming. So, is it possible for correlative protection to differ by population or characteristics? Yes. And so actually, I skipped over that point, and so part of the definition of something being the sort of correlative protection or surrogate marker that means one doesn't have to do more efficacy trials and one can have full confidence in it. Part of the definition would mean that would be that you'll confirm this in more than one population, and actually ideally for more than one vaccine, although that's not always possible. And the reason for that is that despite the analysis I've described, it's possible for something simply to be an epiphenomena. You know, there might be a situation where there's a particular kind of antibody. We measure an antibody in our ELISA. We measure all sorts of antibodies in our ELISA. There's only one particular kind of functional antibody which really is protective. And there might be some populations who don't make that antibody. Or there might be some forms of vaccination which induce what looks like our correlative protection, but actually doesn't induce this special conformation or this special epitope binding that's the correlative protection. So Aisha, you're right to remind me of that additional criteria, that and the more different populations and different vaccination types one can show the correlative protection in, the more confidence one can have in it. Yeah, over. And somebody's asking for the reference for the last slide. So so this example here is one I actually made up. So that's not a real analytical thing, but I will look for that. There are references to the Prentice criteria, and also there are related criteria called the Chin criteria, and I'll post them in the chat afterwards, so that you can see those. Oh, and then there's a question about where does the lower bound of the confidence interval fit into the statistical planning of the trial? So in the non-inferiority study, it is everything. I mean, all of the planning of the power calculation is purely around the lower bound. it does fit into the phase 3 trials. People sometimes say we need to detect a certain efficacy, and they often say, I want to exclude with confidence a certain lower bound. So for instance, if somebody was coming along with a new malaria vaccine now, there will be no point in a phase 3 trial that said, well, you know, we have 40% efficacy, lower bound 10%. I mean, that would not lead to licensure of that as a standalone product. You know, you might license it as a combination product then with other existing agents, but that would be and then that would be a subsequent trial. So so the answer would be yes, the lower bound would fit for a phase 3 trial for new products where you think, well, the only value is going to be in something that's, you know, 60-70% protective and excludes a certain lower bound. That will often be part of the power calculation, and there are ways of feeding that either into a formula, or sometimes people will even do simulations to be confident of what they're excluding. And then Aisha has explained for some pathogens, correlation protection not defined for some serotypes. Is it poor immunogenicity? It's not really poor. It's poor knowledge, really, rather than poor immunogenicity. It's that these correlates of protection. So for something like GBS, where we don't actually have a licensed vaccine, or even a proven to be effective vaccine, people are trying to get correlates of protection based on epidemiological studies. And there will be, there are complexities in doing these things observationally. There are also power issues in the observational studies for some rarer serotypes. And it becomes quite difficult. So those are probably, those I would say are often hypothesized correlates of protection rather than validated correlates of protection. And people have to have varying levels of uncertainty about them. So I think it's restrictions of the epidemiological data that has led to the levels of confidence we have in the different definitions in those cases.
Thank you. I'd make the final remarks before we close.
Sure. Well, look, thank you very much, everybody. Very nice to talk to you all, and I'm very, very happy to be involved in future talks of this sort if there are specific things that people want to pick up on, and I wish you all the best. It looks like I've looked at some of the other YouTube talks. It looks like you've got access to a really great set of resources in terms of developing as vaccinologists, and I wish you all the very best of luck.
Thank you, Professor Philip. We really appreciate having you to do the presentation. Thank you very much to everyone in the webinar. We would like to hear about your experiences and what else you'd like us to discuss in upcoming forums. Before we close, kindly requesting that you take a few minutes to respond to the post-webinar survey posted in the chat. There is a link for that. Have a nice day, everyone. I'm looking forward to more engagements with you. Thank you.
Very good. Thank you all. Thank you. Bye.
Bye.